Hi, my name is Jana Crawford, and this is my Week 2 video lab assignment for genetics. Before mitosis, we have interphase. This is the step where G1, S, and G2 occur. G1 is known as gap 1 and is when the nuclear DNA remains unoccupied and exists as a single normal set of genetic instruction. The S phase, or the synthesis phase, is when the cell copies all of its nuclear DNA, and every strand of DNA is duplicated to create two identical copies called sister chromatids. By the end of this phase, the total number of nuclear DNA has completely doubled. And then in G2, it's known as gap 2 and is when the replicated DNA is scanned for damage and or copying mistakes. The DNA stays intact while the cell builds final proteins and energy supplies needed for mitosis. Finally, we'll start mitosis with the first step of being prophase. This is the step where the nuclear membrane breaks down and disappears during that early stage to let spindle fibers reach the chromosome. The spindle fibers in this example are straws. The reason it breaks down is to give the microscopic spindle fiber a clearer path to attach to the condensed chromosomes and pull them apart. During prophase, this is when the centrosomes that were duplicated during interphase move apart and migrate to opposite poles of the cell. The next phase is metaphase. This is when chromosomes line up at the middle of the cell. Spindle fibers also attach to the chromosomes at their centromeres at this stage. Next is anaphase. This is when sister chromatids separate and are pulled towards opposite poles. Each separated chromatid is now considered an individual chromosome. We're almost done with mitosis, with the next phase being telophase. This is when chromosomes reach opposite poles and they begin to decondense. Two nuclear membranes form around the chromosomes, and I've done this by bringing back the black paper to form the nucleus. This isn't quite when the cell fully separates yet though, that'll be in the next step. Lastly, we have psychokinesis. This is when the cytoplasm finally fully divides the cell and produces two separate daughter cells, each with the same number of chromosomes as the original cell. So what's the difference between mitosis and cytokinesis? Well, mitosis is the division of the nucleus and duplicated chromosomes, while cytokinesis is the division of the cytoplasm and creates two separate daughter cells. Between animal cells and plant cells for cytokinesis, animal cells have a cell membrane that forms a cleavage furrow that pinches the cell into two, whereas plant cells have a cell plate that forms in the center and develops into a new cell wall separating two daughter cells.